So before we get started with the reactions of glycolysis, let's just make sure we know a few important words here. On your what you need to know list, there are these words, hyperglycemia and hypoglycemia. Hyperglycemia just means high blood glucose levels. I don't know, a lot of you are in the health fields and you probably know what blood glucose levels are. Normal blood glucose levels could be anywhere from 70 to 100, but the trick is, do you know the units? You got to make sure you always put units next to a number. You're a chemist this semester, and you're all clinicians, or you're going to be clinicians. You got to put some units, milligrams per deciliter. So normal blood glucose around 100 milligrams per deciliter. So hyperglycemia is when your blood glucose levels are higher than that. They could be 200 milligrams per deciliter, 500 milligrams per deciliter. I mean, blood glucose levels in a diabetic can get really, really high. Hypoglycemia means low blood glucose levels. And low blood glucose levels, we all have low blood glucose when we haven't eaten, if you've been fasting. But clinical biochemical hypoglycemia, we're talking super low. Like it could be maybe 40 milligrams per deciliter, and you can very easily get disoriented and not be able to function properly. So what will prevent hyperglycemia? You got to have the insulin produced. It has to go bind to the receptors and allow the glucose to get into the cells. What will help prevent hypoglycemia? Glucagon. The hormone glucagon elevates blood glucose levels. Glucagon will target the liver. Remember what is stored in the liver? Remember glycogen? Remember how when you put thousands of glucoses together and you form the polysaccharide glycogen, and that's our human starch. It's stored in the muscle. It's also stored in the liver. Well, glucagon will target the liver and force the liver to go through a cascade of events to break down that glycogen, hydrolyze it, and produce glucose and send it into the bloodstream to elevate the blood glucose levels. Epinephrine works in many different ways, but it also has an effect on increasing blood glucose levels. Insulin reduces blood glucose. So I have that over here. All right, so I wanted to just cover that. So now we're going to say the glucose got into the cell. Let's pretend it's in the muscle cell. What is going to happen to that glucose? If you've had a bio class, you've heard of cellular respiration. Basically, you've got glucose, C6H12O6 plus oxygen will give you 6 carbon dioxide, 6 water, and 30 ATP. So this is the general thing that we're going for here. You're going to have a mole of glucose reacting with 6 moles of oxygen gas to produce 6 moles of carbon dioxide and 6 moles of water and 30 moles of ATP. Or you could look at it as one glucose molecule will react with 6 oxygen molecules to produce 6 carbon dioxide molecules, 6 water molecules, and 30 molecules of ATP. How does it do that? Well, it's not one reaction. It's many, many reactions. So the first step is glycolysis. Glycolysis is a 10 enzyme pathway. It's a 10 enzyme pathway, and these enzymes are in the cytoplasm. Remember, we talked about the cell having, the cell having a nucleus and all that liquidy stuff around it. That's your cytoplasm or cytosol. So glycolysis is a 10 enzyme pathway in the cytoplasm. So when the glucose gets into the cell, it can begin glycolysis. Glycolysis starts with glucose and ends with 2 pyruvates. Glucose is a 6-carbon molecule and pyruvate is a 3-carbon molecule. So you end up having 2 3 carbon molecules at the end. All right, so we're going to go through glycolysis step by step. You can look at every reaction in your book so you can follow along, and you need to know what is going on, what the enzyme names are, what the substrate names are, and what the product names are, okay? Structure-wise, you don't need to worry about the actual structures, but, you know, for me, it always helps to draw it out so that I can see what it looks like, but you don't have to worry about the structures, but you should know the names of all the substrates and the products. So the first reaction, glucose reacts with ATP, and you have an enzyme. Enzyme number 1 is hexokinase. Okay, remember what I told you guys about kinase? Kinase is a transferase enzyme. It transfers a phosphate. And a hexokinase works on a hexose. Well, glucose is a hexose. So hexokinase is the first enzyme. The glucose is going to, remember how there's an adenine hooked up to a ribose, hooked up to 3 phosphates. This is what ATP looks like. Go back to the nucleic acid chapter. You can remember the structure of this. So you're going to take this you're going to break this high energy bond. You're going to put this phosphate onto glucose, and you're going to transfer it from this ATP to glucose onto carbon number 6. So the products, I'm going to write the products over here, are glucose 6-phosphate plus what's left over. Well, you took this phosphate, put it here. Now ATP is now ADP, adenosine diphosphate, plus ADP. And that's your first reaction in glycolysis. The substrates are glucose and ATP. The products are glucose 6-phosphate and ADP, and the enzyme is hexokinase. Okay? All right, what happens next? Well, this glucose 6-phosphate is then isomerized. It's an isomerization reaction. So my substrate, remember how I said a pathway, the product of one reaction becomes the substrate of the next one? So glucose 6-phosphate, I'm just going to put a P like this to abbreviate the phosphate, is going to get catalyzed by and isomerized by an enzyme called phosphoglucoisomerase. Okay, so that's the name of the enzyme, phosphoglucoisomerase. It's an isomerization reaction. You're just going to turn glucose and fructose are isomers. So you just manipulate some things around and you end up with fructose 6-phosphate. All right. So the second reaction in glycolysis takes glucose 6-phosphate, isomerizes it, and turns it into fructose 6-phosphate. All right. The next step, we're going to take this fructose 6-phosphate and do something with it. Remember, the product of one reaction is the substrate of the next one. Out here. So fructose 6-phosphate. We're going to throw in another ATP. Here's another kinase enzyme, because this kinase is going to take another phosphate group from another second ATP and put it onto carbon 1 of the fructose. So fructose 6-phosphate plus ATP. The enzyme is phosphofructokinase. Okay. Again, the name of the enzyme helps you figure out what is going on. It's a kinase. That means we're transferring a phosphate group from an ATP. And what are we transferring it to? To a phosphorylated fructose. So this process of transferring a phosphate is a phosphorylation, and the category of enzyme is the kinase. All right, so fructose 6-phosphate plus ATP The enzyme is phosphofructokinase. The product is going to be fructose 1,6-diphosphate or bisphosphate. I think your book calls it bisphosphate, if I'm not mistaken. Honestly, I don't usually use the word bis. I like to use di, but I guess if your exams have biz, it's probably good to use the biz. Bis means di. All right, so fructose 6-phosphate and ATP are the substrates of this third enzyme. The third enzyme is also a kinase. It's also a phosphorylation. So the first enzyme and the third enzyme are these phosphorylations where you're taking a phosphate group from ATP, putting it onto a substrate. And what's left over is ATP already removed a phosphate, so what's left is ADP. So these are the products. Okay, so those are the first 3 steps of glycolysis. In the next video, we'll continue on with the rest of glycolysis. Okay.